Hello, my name is Dr. Adam Smith, and I'm going to give this presentation entitled Conventional to Unconventional Cytogenetics, New Techniques for Structural Genomics. I have no conflicts of interest to declare. I like to often show this cartoon as it makes an interesting point about how for a long time people have been predicting the end of cytogenetics. And as a variety of new techniques were developed, whether they be blotting techniques or PCR-based techniques, at every point we have heard that this sounds the death knell for cytogenetics, especially so in the era of microarray and NGS sequencing. And in some aspects, it's true that parts of cytogenetic testing have been replaced by other more effective methodologies. However, in the large part, the field of cytogenetics has remained a powerful technique over the last 60 years, where it's been used both clinically and in research. So we ask ourselves the question, is cytogenetics dead? And I believe this is actually the wrong question. What we're actually talking about is as the technologies to look at what we're actually looking at in cytogenetics, which is the structure of the chromosomes and how those structures are rearranged in health and disease. We're getting better techniques and better technologies to look at those things, and it becomes a question of, maybe we're changing the techniques a little bit, but we're still studying the same thing. So with that, I present to you the original whole genome. And the karyotype still has an immense amount of power in terms of an analytical technique. It's relatively quick to complete. It costs very little from a reagent standpoint. And it gives a cell-by-cell analysis of the structural composition of the human genome. So you're able to look at each chromosome. You're able to look at how and whether or not there are large structural rearrangements that are visible. And quite often, some of these rearrangements that occur in regions that are repetitive, that are not amenable to detection by most molecular techniques that are available today. Which is why in cancer cytogenetics, for example, the karyotype is still a very powerful tool. And the tool in which a lot of diagnostic and prognostic systems are developed for a variety of different hematological and lymphoid diseases. So how has cytogenetics already been affected by change? So we've seen changes occur, whether it be in terms of the progression of prenatal testing, for aneuploidies and how, for example, aneuploidy or FISH testing for aneuploidies replaced karyotype analysis for a rapid result, which was in a lot of places replaced by QF-PCR. And for a lot of people now is being replaced by NIPT or NIPS, this non-invasive type of testing of maternal blood that we can detect changes in the fetal DNA. Another big disruptor of cytogenetics in terms of technology shifts has been the microarray. And here pictured is the process of making an Affymetrix microarray using a process known as masked photolithography, where probes are actually created on a chip, one base pair at a time, using a mask and using chemistry, which allows you to add bases in a sequential order. So this process enables you to create hundreds of thousands to millions of probes on a single chip and then interrogate the genome. And so we did some work at the BC Cancer Agency when I was there several years ago, looking at comparison of karyotyping studies to array studies in both AML and MDS cases. And you can see here, this is a karyotype study that was performed with a fairly complex karyotype. Also performed microarray studies on this. Now, one of the things that I did want to point out very briefly was this topic known as chromothripsis. So just before we go on, I'm just going to take a brief segue here to talk about this. Now, chromothripsis is a type of rearrangement that occurs when there's a catastrophic problem in terms of the cohesion of a particular chromosome. So you have an original chromosome sequence like this, and then you get catastrophic breakage where there's many double-strand breaks and the chromosome is actually broken into many pieces. And then using a variety of repair mechanisms, the cell will try to put the chromosome back together. But it doesn't necessarily put it back together exactly like it found it, of course. It can put it back together in a slightly different order. Some pieces can be lost, some pieces can be gained, and you have this significantly altered chromosome that occurs after this catastrophic. So I point that out because when we look at that karyotype that I just showed you, this is the metaphase cytogenetics here shown at the bottom, you can see that there are a variety of these rearrangements that were detected. So again, you can see the plus X called very easily. We've got the deletion on chromosome 3 that was quite small, called by metaphase cytogenetics, also seen by microarray. You've got the deletion 5q, which is a very common abnormality in myeloid neoplasia. The plus 10, of course, wasn't called. This is, of course, a composite karyotype, so likely at low level and below the limit of detection for microarray. And then we had a bunch of stuff that we knew was going on here. We used these, some of our favorite terms in cytogenetics for G-bending, the add and the question mark and the marker, as indicating items of ambiguity in terms of analysis, where we're not able to accurately identify what's going on. And so when we look at those chromosomes involved, we look at this region of chromosome 11, where you can see a deletion, where you can see a gain. Again, on chromosome 12, you can see deletion, gain. And then down here on chromosome 14, you're seeing many segments of both gain and loss, probably evidence of some kind of chromothripsis and rearrangement. And then again, you've got more rearrangement on chromosomes 15 and 16. And so you see that very clearly. So comparing these two technologies, the karyotype versus the microarray, they have different advantages and disadvantages, obviously. The microarray cannot determine structure. So even though you can see gains and losses, you don't necessarily know how those gains and losses go together. As you know, they can't see balanced rearrangements, obviously. And it's difficult to differentiate between subclones and or unrelated clones. So you could have two clones, for example, that have an abnormality in about the same percentage of cells. And from the microarray's perspective, they could be in the same clone or different clones, and you would never know the difference. On the other hand, the karyotype has some limitations as well. So there's the resolution issue with karyotyping. And generally in bone marrow karyotyping, you're looking at probably around 20 megabases as a sort of standard cutoff for resolution. So when you have rearrangements that are below 20 megabases in size, and for this we're talking about deletions and duplications largely, you're not going to be able to see them. Obviously, of course, translocations, they're quite small in terms of the fact that material that's actually moved, but this applies to them as well. So when you have a translocation, for example, that is very close to the end of the chromosomes, and you may switch a small amount of G light material or G dark material, for example, you may not be able to see that rearrangement. Karyotyping is, of course, dependent on the presence of a sufficient number and good quality metaphases for analysis. And resolving more complex and especially cryptic rearrangements can often be difficult. So again, in some ways, these techniques can be complementary, and they can do certain applications better than others. And again, it depends again on the answer that you're looking at. And then we have to talk a little bit about next generation. So next generation sequencing is an umbrella term. It covers a lot of different strategies. There's a lot of different companies that have different ways of interrogating the sequence. And this isn't only in the actual reading of the sequence itself, but in terms of the how to amplify the template to before you even read the sequence. There's a bunch of different strategies, and these are basically divided into a few different categories. And so emulsion PCR is common to the 454, Roche, the Solid, Gene Reader, Ion Torrent platforms, where you basically have a little micro ball here with primer on it in an emulsion of oil, and it forms these little micelle droplets. And then again, you put your templates in, you get hybridization and amplification, and then you can amplify out your targets. And so you should get a single sequence, ideally amplifying on all the little primer pairs on an individual ball inside a droplet. Solid phase bridge amplification, this is the Illumina technique, basically where you've got a bunch of different little primer or adapter molecules on a flow cell, and then you flow the template that has been prepared with these adapters on it, and then it undergoes what's known as this bridge amplification. So the molecule with the area that you want sequenced in the middle bends over and finds a complementary primer sequence on the flow cell, and it then makes a copy of itself. And then the two daughter strands separate, they stand up individually, fold over again, find another complementary primer, duplicate, and you go through many cycles of this so-called cluster generation, and then you get 100 to 2 million clonal clusters. And then each of these clusters can be read using fluorescence. You've got some other techniques like solid phase template walking, which is less common, and of course this in solution DNA nanoball generation, which is the BGI, or the Beijing Genomics Institute solution, where they have these adapters and then they circularize these DNA templates, which can then be cleaved, and then you can use this iterative ligation, where 3 additional rounds of ligation, circularization, and cleavage generate a circular template with 4 different adapters, and then you can do this rolling circle amplification to basically create a lot of template, and then hybridize this nanoball to a patterned flow cell. So all of the sequencing by synthesis techniques work essentially the same way. And the idea is that you have a template molecule, you have a primer, and then you have a labeled nucleotide that gets added in a stepwise fashion. So these terminally blocked nucleotides are added with the fluorophore, the corresponding complementary DNA base pair. At each phase, these are then imaged, and so each clonal cluster can then be imaged, and the color can be red, which represents which nucleotide has been added to this growing template. The blocking agents can then be cleaved and washed, and then a new cycle can be begun. So after many of these cycles, you can generate hundreds of base pairs, potentially, of sequence. And again, in a very similar fashion, the QIAGEN gene reader works in a similar way, using these little balls that were generated by the emulsion PCR, and then in the same way, the nucleotide addition and imaging takes place. So using these techniques have been fruitful for research into a variety of different diseases. And in fact, looking at the number of cryptic genomic lesions, for example, in a recent publication by our group at Princess Margaret, has shown that sometimes the conventional cytogenetic analysis that we do can have its limitations, obviously. for a variety of reasons, and some of those are illustrated by these cases here. So for example, we have two cases where by conventional cytogenetics we called a translocation 1217, which can be seen here pretty clearly. However, when a combination of next generation sequencing using whole genome sequencing and RNA-seq was performed, it was discovered that this was in fact a more complex rearrangement with a very small segment terminal rearrangement involving chromosome 11 and 12, and then chromosomes 12 and 17. So we did get part of the rearrangement. So below here you can see all of the derivative chromosomes. So you can see you have a derivative 11, a derivative 12, and a derivative 17. And so this is a very interesting use of this technology in order to show some of the limitations that we face by doing a conventional analysis. Similarly, in case 6 here, we called it T1112, P13, P13. So again, this is terminal segment exchange of chromosome 11 with the P arm of chromosome 12, showing here chromosome 17 for your information. However, we did not call it a rearrangement of chromosome 17. As again, you can see in these bone marrow karyotypes, the resolution and banding is not the best. But again, what we're seeing here is a chromosome 11, chromosome 12, chromosome 17 rearrangement. And in fact, this is a fairly complex rearrangement where you're seeing multiple segment exchanges between chromosome 11 and 12 and 17. In addition, you're seeing a loss of several segments down here as well. So this is a complex rearrangement, the complexity of which was not appreciated by conventional cytogenetics. That being said, if we look here at the same cases again, and we look at the supplementary information here from this paper, looking at the concordance between the individual cytogenetic abnormalities and concordant NGS findings, there is in fact a high level of concordance in the abnormalities. So you can see everything basically in red is concordant between the NGS and karyotypic findings. There are, of course, some things that are not concordant. So, for example, the isochromosome 17q was not detected by any NGS method. Again, this being a composite karyotype, it's unclear if the level of this rearrangement was sufficient enough for it to be detected. And so in several cases, we can see similar types of rearrangements that were detected by both methods. Again, some of the ambiguity with the banding, with the calling the band designations in cytogenetics can often be difficult. And then if you see here, for example, the actual breakpoints called by the NGS software, where you've got a P12.3 and a Q14.2 breakpoint. So close, but not exactly there. So again, the use of both of these techniques together is extremely powerful for looking at the structural rearrangements that are present. So genome sequencing, of course, is a relatively new technique in the sense that the short read sequencing of the modern generation. It's now used at massive scale, so there are more than 100,000 genomes. Short read sequencing is incomplete, however. There's, again, lots of caveats when using a short read sequence. Repetitive regions are unmappable. Highly variable regions are difficult to resolve, such as the HLA genes. Non-reference sequences are ignored, and genome assemblies are fragmented and incomplete. So to completely sequence human genomes, we need much longer reads. And so there's a couple of key platforms, the Pacific Biosciences and the Oxford Nanopore platforms, that are able to actually natively perform long reads. And then using some chemistry tricks, a variety of other platforms are able to perform what's called synthetic long reads. So what is exactly the difference between short read and long read sequencing? So if you look, for example, at the structure of a hypothetical gene here, you've got various exons and some intronic spaces. And you've got short reads. And so you've got all of these short reads that, and for the sake of argument here, we'll just say that these short reads are randomly dispersed among the exonic and intronic sequences, as opposed to, say, what you would do in a panel test where you would concentrate your pull down or your baits over the exons, for example. But you can see that you've got a lot of reads here and that they aren't connected to one another. However, with the long read sequencing, you've got a much longer read length. The breaks are much less common, and so you've got much more sequence that ends up being overlapping. And so what does this help with? So it helps with a bunch of different things. It helps in terms of trying to figure out what's going on in terms of some types of structural variation. So when you've got inversions, potentially, of particular genes or introns, or you've got gene conversion events, for example, or you've got mobile elements, for example, that might move around. You've got more ability with a longer read to detect these kinds of things. You're also able to handle repeat expansion more easily because you're able to read through the entire length of a repetitive sequence and come out on the other side, so then you can align to the unique reference sequence and figure out actually how many repeat elements you have. Phasing becomes a possibility. Phasing with short read sequencing is sometimes possible depending on if there are multiple SNPs present and you're able to actually figure that out, but it tends to be fairly rare that it can actually happen. But with longer reads, it becomes possible because you have these long sequences, these long reads, where you have multiple SNPs that you can then use to align and phase out the reads into maternal and paternal. Pseudogenes is another area that becomes incredibly helpful. So you've got many genes, for example, in the genome that have a duplicated copy that is often non-functional, or perhaps maintains some functionality but not at the same level as the original gene. And usually these have very small sequence variations. But using a short read platform, it's often very difficult to tell the actual real gene from the pseudogene. And so using longer reads, you're able to read through the entire sequence. You're able to find unique sequences that flank these regions, and then you're able to basically look at if there are any changes specific to this gene or the pseudogene. So this was a really excellent review paper looking at a lot of the current next-gen sequencing technologies by Sarah Goodwin, John McPherson, and Richard McCombie. It is an excellent review of all of the, or most of the techniques that are coming out and what is essentially happening in the field. It deals with a lot of the different issues around how a variety of different platforms deal with labeling of template in order to do detection. So looking at microarray labeling, qPCR labeling, nanostring labeling, and of interest, labeling for optical mapping. So optical mapping is a technique that we will discuss a little bit later, but I just wanted to very quickly point it out that you have this technique where you're going to label with a fluorophore at a particular site, a particular 6 base pair sequence that you can then run out and have a particular arrangement of labels that can specifically identify sequences where they map to in the genome. So for long read sequencing, you've got the two main systems, the Pacific Biosciences technology and the Oxford Nanopore. And so the Pacific Biosciences is using this Smart Bell template with the 2 hairpin adapters, where you've got these adapters on either side and your template sequence in the middle. And the sequencing takes place on these ZMW wells. And so these labeled nucleotides move through, and in fact there's a modified polymerase that's basically at the bottom of these wells, and as this template goes in, it can then be read by this polymerase And as this happens, a camera can record the emitted light of the incorporated nucleotides, and then you can generate a fairly decent length of long read sequence using the SmartBell template with this specific well. Oxford Nanopore, on the other hand, uses a pore-based sequence where the entire strand of the DNA sequence actually passes through a pore. And depending on the displacement of the pore, you end up getting a change in the current. And that current can then be translated into a particular sequence. Just talk about that a little bit more later. In terms of some of the synthetic techniques for doing long reads, Illumina has some techniques for long read sequencing where you have a long DNA sequence that's fragmented down to about 10 kilobases, that you then have all of these molecules per well. They're cleaved, DNA is barcoded and fragmented to 350 base pairs, which is the approximate length that they're sequencing using a short read sequencer. So then then barcoded, so the DNA from the same wells shares the same barcode. It can then be pooled and sequenced on a standard short read sequencer. 10x Genomics is another company that is using an emulsion PCR-based method. So it takes arbitrarily long DNA mixed with beads loaded with barcoded primers, enzyme, and DNTP. And then it creates these gems. And so each micelle inside the tube here has one barcode out of 750,000. Long fragments are amplified such that the product is a barcoded fragment of approximately 350 base pairs. And then this pool, the emulsion is broken and the DNA is pooled, and then it undergoes a standard library preparation. And so you end up with these linked reads. So all the reads from the same gem derive from the same fragment, thus they are linked. And the reads are dispersed across these fragments, and so no single gem will achieve a full, will actually end up being read fully. But what you end up with is by dispersing across the different fragments, you end up with complete sequence across the entire region, just using all of these different GEMs. Going back to nanopore sequencing, it was initially patented back in 1995 when it was noted that the individual the current changes that could occur when you were able to pass bases through these nanopores. And then what it was also noted, as I said before, that depending on the nucleotide that passed through, the size of the nucleotide, and all the nucleotides are slightly different sizes, of course, and then depending on how much displacement occurred in the pore, you could measure this change in current. It gave a specific signal in terms of the current that was changed when the nucleotide passed through. And so this technique has been obviously worked on significantly over the past 20 years or so in order to improve it so that it can be actually used to sequence. And so one of the initial problems, of course, with nanopore sequencing was a fairly low accuracy. And a 70% accuracy when you're talking about sequencing hundreds of thousands to millions of bases of DNA is a particular problem. And so as the chemistry and the base calling algorithms have improved, the accuracy has also improved. So on the current pores and algorithms, we're now getting a raw accuracy of 90% approximately. And with some additional tricks, this accuracy can be improved even more than this. So this is what the Oxford Nanopore with their instruments looks like. This is a standard USB cable. It's a very, very small instrument, the MinION. It can do quite an impressive amount of sequencing. It's very mobile, so you can see how field applications potentially or right in the office sequencing could be done, despite the fact that it would take considerable time still. GridION is basically a slightly larger version, higher capacity, where you put a bunch of min ions basically in the single machine. You can do sequencing like that. So there's a variety of throughputs and outputs for these machines. The interesting thing about the long read sequencing has always been the contig NG50. So what's the sort of average size of a contig that can be developed? And so this is a human genome build that was done in January 2017. The average contig NG50 size is about 3 megabases, 30x coverage with a polish accuracy of 99.1%. And so you can see that you're still seeing a lot of segmentation. You're still seeing a lot of different breaks. Interestingly, if you know anything about cytogenetics, you can see some interesting characteristics of the chromosome. So you can see regions of facultative heterochromatin here on chromosome 1, chromosome 9, also on chromosome 16. You can see, for example, very gene-rich G-light regions up here on the P-arm of chromosome 1. So you can see a lot of the sort of mirrors of chromosome banding here by this technique. However, in order to really get good coverage, read through a lot of the very long repetitive sequences in the human genome, you need a very long read. So I just showed you some data with an NG50 of 30 megabase pairs. So in order to span a repeat of 500 kilobases, you really need you start to get into much higher levels of NG50. So moving on, to get these ultra long reads, you need a technique that preserves the DNA without breaking it up too much. A lot of current DNA extraction methodologies, they're not particularly gentle in terms of, when the DNA you pull out, it's already pretty highly fragmented before you go ahead usually and fragment it some more. So this careful preparation can achieve greater than 100 kilobase read length at N50. So with some very, very long reads potentially at 2.3 megabases. So looking again at a May 2018 build of the genome, again you can see now more large contig coverage, that NG50 at 10.3 megabase pairs with accuracy up to 99.77, improving dramatically. And finally, the Human Genome 2019 with an NG50 of 75 megabase pairs. We're starting to see very, very long chromosome arm level coverage where we were able to read through these sequences. We're able to read through region, not quite yet through these regions of highly repetitive heterochromatin, but certainly through centromeres and a variety of other sequences. So the case for nanopore sequencing in clinical applications is obviously quite useful potentially to have this long read sequencing. A lot of translocations, of course, can be precise, so they have a precise breakpoint in a unique sequence region, so they're relatively easy to read through. However, you can get rearrangements that are not precise, that are not gene fusions, that are still important, like the MYC-IGH rearrangement, for example. And this is important to know. The other thing that happens, of course, in cytogenetics that's sometimes unclear is we can detect a particular rearrangement. So, for example, we might, by looking at the G banding, look at and say, oh, we see something with chromosome 10 and we see something with chromosome 11. We're not quite sure what's going on, and we don't know the exact breakpoint, so we're unable to say, this is a rearrangement that involves a particularly important leukemia genes such as KMT2A, formerly known as MLL. So we did some chromosome painting. We had some G-banded cytogenetics. You can see the segment exchange here between chromosomes 11 and 10. However, in order to actually identify the MLL-T10 KMT2A rearrangement, we would either have needed a specific FISH probe for this, or in this case, we were able to do this with the help of Jared Simpson and the MinION and doing some sequencing. Now, the MLL-T10 KMT2A translocation is a very interesting translocation. The translocation is such that it's not a single break and join translocation. And the reason for this is because the MLL-T10 and the KMT2A gene are on actually different strands. One's on the plus strand, one's on the minus strand. So if you have two double-strand breaks and a rejoin, you can't actually get an in-frame fusion protein. So in order to do this, what you actually need to do is you need to have an additional break to flip one of the segments around to get this translocation product in frame. And in fact, this is what you have here. You have got two rearrangements. You've got a rearrangement involving 11q23 and chromosome 10, and another one with 11p. And this is able, this multiple rearrangement is able to get this particular translocation in frame. And it was great to be able to detect this using this technology. Of course, this is not a good prognostic translocation generally, and so the patient did relapse. We had another sample at relapse. We're able to see the original diagnostic translocation here, plus some additional translocations that were present post-relapse. And again, you can see here, this is the chromosome 11 plot, where the breakpoint exactly is inside the KMT2A gene. Now I'd like to discuss briefly this relatively new technology called optical mapping. So optical mapping is not exactly a sequencing technology, but it does share some similarities with sequencing. So basically the process is like this. You start with a DNA extraction of extremely long DNA molecules, and for good alignment, you're looking probably for fragments above 100 to 150 kilobases in length. You're then going to label that DNA at a specific sequence motif that repeats throughout the genome with a relatively regular frequency. And then you put it on the Sapphire instrument from BioNano. And what that does is it goes through a bunch of nanochannel arrays, and it takes this DNA and it strings it out into a very, very long linearized fragment. And then basically it generates an image like this of all these linearized fragments that are labeled with this repetitive sequence, which can then be aligned to a reference genome. So using this technique, it's then able to look for gains and losses in sequences and copy number change. It's also able to detect balanced rearrangements such as translocations or inversions based on the change in the sequencing pattern. So it's an extremely versatile technology in order to detect these larger structural type variations at a relatively high level of resolution. Much higher level of resolution than, say, a G-banded karyotype, but maybe not at the level of resolution of an individual, the individual nucleotide level like you would get with sequencing. However, the cost is dramatically less than a sequencing platform to get genome level structural information, and it's a really good platform for looking at the kind of structural variation that we tend to look at in cytogenetics. It's also quite versatile in a lot of ways, in that some types of variation that can be missed by short and long read sequencing can actually be detected using the BioNano platform. So you can see that in this example where there's a complex rearrangement involving a translocation between chromosomes 7 and 10, and in this same region a 1.61 megabase deletion and a 210 kilobase inversion and a 399.9 kilobase pair deletion, that all of these rearrangements were able to be detected with the optical mapping platform. And so this is one of the great strengths of this platform. In order to be able to look at this variation in the complexity, it reminds me of the similar case I showed earlier from our data, where we had showed that a much more complex rearrangement occurring. And I think this is a general theme that we're going to see as we get better resolution when we look for structural variation, that the straightforward translocations and dels and dupes that we are detecting using conventional cytogenetics, as we look a little bit deeper, we're going to start to see more structural variation and more complexity in that variation. So just to talk a little bit about all the different platforms that are out right now, and this paper that was recently published in Nature Communications, looking at haplotype resolved structural variation in human genomes. And the idea here being that they looked at a bunch of different platforms, Pacific Biosciences and Oxford Nanopore, as the native long read technologies. Illumina short inserts, this is your standard libraries. Then this is a long insert WGS library, a jumping library. These are synthetic long read libraries. Your 10x Chromium, which is the micelle based GEM method for developing longer reads. BioNano here, TrueSeq SLR, StrandSeq HiC, and a bunch of different platforms that are being used to look at copy number gains and losses and structural variation. And so depending on the platform, they obviously have a variety of strengths and weaknesses. They're going to detect a lot of things. And depending on the platform, of course, the sizes of those things is going to change as well. So, and then you can see that they've done basically a bunch of different cell lines here. They've done all the different platforms. They've looked at deletions. They've looked at insertions. Depending on the platform, it's quite remarkable to see the difference in terms of the calls. Some of these calls are kind of straightforward to understand. Looking at sort of say PacBio or Illumina, which is giving you sequence level changes. And you can see here by the average length, where BioNano is calling a lot less, for example, but with a much higher average length. So we know for a fact that the BioNano platform has a lower limit of resolution, and in fact is probably at about this level and certainly nowhere near this level. So PacBio is calling a lot of deletions that's below the level of the BioNano system. And then, of course, the problem becomes interpreting all of this variation and what to do with it. So some work done here by James Broach at the University of Pennsylvania, published recently. This paper, an integrated framework for genome analysis, reveals numerous previously unrecognizable structural variants in leukemia patient samples. And interestingly, looking at polymorphic and somatic structural variants by a combination of optical mapping and WGS. And so again, we're looking at gains and dupes, deletions and losses, insertions, inversions, and interchromosome translocations. And so, of course, these are categorized into either somatic or polymorphic. And so, interestingly, right away, you can see that there is an enormous number of polymorphic gains, losses, insertions, and inversions that are occurring in all of these samples that are tested. and a much lower number of somatic changes. Of course, somatic deletions and somatic gains, inversions tend to be, the somatic inversions tend to be much less. And of course, obviously right here, the interchromosomal translocations, very, very few polymorphic interchromosomal translocations detected, and a lot of somatic ones. You would potentially expect to see, if you did more samples, a few interchromosomal translocations that are polymorphic here, but the great majority, of course, are somatic. And so it's a very interesting, it's very interesting work that's been done, looking, for example, at patients' PBMCs. You've got your B lymphoma cells here, your normal T cells. You can then activate culture, stimulate T cells. You can sort out those T cells. And so you can take the circus plot looking at the structural variation occurring in the B cells, and you can compare it to this baseline, or should be germline the rearrangements that are occurring in the circus plot here. And again, you can see that in the B cell lymphoma, you can see evidence of lots of rearrangements, translocations, copy number changes, and of course in the T cells, no translocations and a much more stable genome, of course. This is an example of what a karyotype in one of these cases might look like, with a variety of structurally visible abnormalities, including deletion or translocation 4-5 here. You've got a monosomy 7, you've got an AD8P23, monosomy 12, and this deletion of chromosome 1. And interestingly, if we look at the average number of SVs per sample, you can see that for gains, losses, insertions, inversions, and intrachromosomal translocations, generally this is pretty low. The minute we start going towards using a combined approach, or even just either of these on their own, you're starting to see fold increases in the number of detections of gains and losses, insertions, inversions, and even translocations, above what was detected using conventional cytogenetic studies alone. So interestingly, this sort of the place for this type of technology in the clinical approach to studying cancer, I think, is going to be a very interesting area of research and development that is ultimately probably going to replace a lot of conventional cytogenetics in the next few years. So let's talk a little bit about clinical utility. In order to talk about clinical utility, we need to think about some of the things that are important in the clinic, and why some of the technologies and assays that we use are used today. And really some of the key factors are, of course, turnaround time, cost, and analysis. How complex is it to analyze the data? What info can be provided? What information do we have about the particular abnormalities? So as we get towards tests that provide more data, provide more information, what do we know about those things? Are we just basically doing information overload, and we're not actually providing useful information that can be used to help treat patients? From that review paper that I mentioned a few slides back, they have a really interesting table that compares a lot of the different sequencing platforms, both historical platforms that don't get much use these days, such as the 454 systems and the old PGMs, which aren't used very often, to some of the newest and best sequencers on the market, including HiSeqX and the long-read sequencing platforms like the Pacific Biosciences RS2, the Sequel, the MinION, the Prometheon, etc. And so, of course, the interesting point here from a structural genomics point of view, from a genome point of view, is, of course, this cost per gigabyte. Cost per gigabase. And so you can see that some of the costs per gigabase are in fact really quite high, even on the synthetic long-read side and the core-based sequencing. The cost per gigabase is still relatively high. And in order to make these viable techniques from a clinically useful perspective, where they could be used on a lot of patients and not just a very limited subset, it's important to look at this particular cost. And so when you look at something like the Illumina HiSeq X and the cost to do a genome here at about $7, so you're talking about a cost in around $500 to $1,000 potentially to do a genome. And this is getting down to the kinds of costs that we need to get to in order to make doing structural variation testing by sequencing a reality. And the other interesting part to look at too, of course, is the runtime. The runtime is a really interesting look at when you look at some of the different systems and how they deal with sequencing, how long it takes to get through. And so here on the HiSeq X, probably less than 3 days. It's still a pretty long time if you're in a situation where you have a relatively tight turnaround time and you need to give an answer. That's potentially problematic. Certainly on the HiSeq 2500 version 4, where you have turnaround times or run times of 29 hours, for example, you're starting to get into the kind of time that would be necessary in order to give a reasonable, to get data analyzed, potentially to give a reasonable answer in time. And if we look at an example of a testing workflow, for example, at the University Health Network, where we have an integrated testing of a sample, let's say of a bone marrow sample that comes in for acute myelogenous leukemia, we have a bunch of tests that are going on in parallel at any given time. So we've got flow cytometry and morphological analysis that are happening right off the bat that generally within 24 hours we've got preliminary diagnosis, we've got flow cytometry data, so we've got a confirmation of diagnosis. We've got molecular and cytogenetics testing in progress, and depending then on the test, there's a variety of different turnaround times. So 5 days for FLT3 NPM1, up to 21 days for the myeloid NGS panel. For certain subtypes of AML, such as APL, AML M2, M4, that have specific rearrangements, there are qPCR-based assays, again, that have fairly rapid turnaround times. And of course, karyotyping and FISH and cytogenetics with an average 5-day turnaround time. So this is a pretty rapid turnaround time that's required for testing. So in order to make any kind of technology useful in the clinic, these kinds of turnaround times need to be taken into account. Certainly on the NGS side, getting this number down, there's a lot of variables to work on to get things reduced. DNA extraction is one of the variables. Library preparation, sequencing time is potentially a big variable. The bioinformatics pipeline, your variant interpretation and reporting, all of those elements take time in the reporting workflow, and so getting the turnaround time shortened is a pretty substantial task in terms of trying to reduce some of these elements. So in conclusion, what do we want to say about the conventional testing paradigm and what we've discussed and explored today? Certainly, there's lots of new tools for looking at structural variation, lots of new sequencing platforms and variations on techniques that are allowing more structural variation to be looked at with a higher degree of accuracy. More complexity is being seen in the genome than was detected and is able to be detected just by short-read NGS, due to the variety of limitations that the short-read NGS platform has. And ultimately, from a cost perspective, to replace conventional karyotyping, the key issues that I just discussed, TAT, cost, and analysis, there's still a lot of work that needs to be done to really take that conventional karyotype out of the equation and to replace it with other technologies. Certainly, as the technologies especially such as optical mapping, get better and better, we're more likely to see um, these kinds of techniques be used to replace conventional karyotyping, as they offer greater resolution and the ability to detect breakpoints and rearrangements with a higher degree of accuracy than are currently available using conventional cytogenetics. And that's all that I wanted to talk about in this presentation, and thank you for your attention. Oh, as final, some acknowledgements. People who helped and collaborators that we've worked with on the projects presented. I'd like to just acknowledge Dr. Jared Simpson from the Ontario Institute for Cancer Research, Dr. Tracy Murphy, Mark Minden, Jae-Sun Kim, Princess Margaret Cancer Center, Dr. Ali Carson at the British Columbia Cancer Agency, all of the cytogenetics and genome diagnostics lab staff at the University Health Network, and Dr. Elena Kolomietz at Mount Sinai Hospital. Thank you.